Section: Introduction. In this section, we introduce the core philosophy of artificial life, A-life, which encourages us to explore not just life as we know it, but also life as it could be. Our focus is on using computational simulations to investigate a wide range of possible scenarios rather than just one. This allows us to understand why different simulation setups lead to various emergent behaviors. For the first time, we aim to automate the search through these simulations using foundation models from AI. Although the mechanisms of evolution and learning in A-life are diverse, a significant challenge has been the absence of a systematic way to explore all possible simulation configurations. Without this, we often rely on intuition to create the essential rules of an artificial world. The complexity of interactions among simple components can lead to unpredictable outcomes, making it hard to design simulations that demonstrate self-replication or ecosystem dynamics. Consequently, we often end up with simulations that are manually crafted for expected results, which limits our chances of making unexpected discoveries. By automating the search for simulations, we can shift our focus from guessing rules to articulating the phenomena we want to see emerge. However, defining these target phenomena is itself a challenge, which has hindered automated searches in the past. While there have been attempts to quantify A-life through various metrics, these often fail to capture the complexity we seek. We propose a new approach called Automated Search for Artificial Life, ASAL. In this framework, we start by defining a set of simulations, known as the substrate. ASAL offers three methods for foundation models to identify interesting A-life simulations. 1. Supervised target searching. This method looks for simulations that produce specific events or sequences, helping us discover new worlds. 2. Open-endedness searching. This method seeks simulations that generate ongoing novelty, leading to worlds that remain interesting over time. 3. Illumination. This method finds a variety of diverse simulations, revealing unique and unfamiliar worlds. We demonstrate the effectiveness of ASAL across various A-life substrates, such as boids, particle life, and neural cellular automatas. In these cases, ASAL uncovered new lifeforms and expanded our understanding of emergent structures. For instance, it identified unique flocking behaviors in boids and new self-organizing cells in Linnaea. Additionally, ASAL allows us to quantitatively analyze phenomena that were previously only qualitatively assessed, providing a more human-like way to measure complexity. Importantly, ASAL is flexible and can work with different foundation models and simulation types, making it a valuable tool for future A-life research. To our knowledge, this is the first effort to use foundation models to drive the discovery of A-life simulations. In the following section, we discuss the motivations behind A-life. This field studies life through artificial simulations, focusing on the general properties of life rather than just the specific forms found on Earth. A-life encompasses a wide range of systems, from cellular automata to neural network agents, and emphasizes emergent phenomena like self-organization and collective intelligence, which have also influenced AI research. Section Summary. In this section, we introduce the concept of Automated Search for Artificial Life, ASAL, which aims to automate the exploration of simulation configurations in artificial life research using foundation models. By enabling methods such as supervised target searching, open-endedness, and illumination, ASAL facilitates the discovery of novel lifeforms and emergent structures, ultimately enhancing our understanding of complexity in artificial life systems. Section: A-life substrates. In this section, we explore various substrates used in artificial life, A-life, to investigate different phenomena, ranging from chemistry to social structures. Early on, we relied on Conway's game of life and similar cellular automata to understand how complexity can arise from simple local rules. Linnaea expanded on these concepts with continuous dynamics, leading to new variations like flow Linnaea and particle Linnaea. Neural cellular automata, NCA, took this further by using neural networks to model continuous cellular automata. Instead of a 2D grid, particle life employs particles in space that interact to form dynamic patterns. Similarly, boids simulates the flocking behavior of birds and fish. Other structured substrates, like Biomaker CA and JaxLife, focus on the behaviors of plants and societies, alongside notable systems like Evolved Virtual Creatures and Neural MMO that study survival in natural settings. Some substrates even aim to explore unique phenomena, such as self-replicating programs. All these systems are designed to investigate specific life aspects, emphasizing emergence from simple components. Our approach is substrate agnostic, as long as the substrate can be represented visually. Most A-life substrates are designed to be interpretable, including those that are not visual. We also discuss the role of automatic search in A-life, which has proven useful when outcomes are clearly defined. Initially, we used genetic algorithms to evolve cellular automata for specific computations. More recently, we have focused on objectives related to linear, such as speed, to discover new organisms. Biomaker measures agent survival over time, while NCA aims to match a final state to a target image. Novelty search, inspired by A-life, requires a good representation space to be effective. Map Elites searches along two predefined axes of interest, and intrinsically motivated discovery explores representation spaces to find new self-organizing patterns. Linear Breeder employs Map Elites to find organisms with specific traits, although it has limitations in guiding searches. Additionally, training an autoencoder solely on substrate images may not yield representations aligned with human understanding due to insufficient data diversity. We also address efforts to quantify emergence and complexity. In information theory, Kolmogorov complexity assesses the shortest program that can create an artifact. Instead of directly measuring artifact complexity, sophistication evaluates the complexity of the set to which the artifact belongs. Assembly theory, rooted in biochemistry, aims to quantify evolution by counting the minimal steps needed to assemble an artifact from basic components. However, these metrics often fall short of capturing the intricate human understanding of complexity. Many complex systems exhibit computational irreducibility, meaning their emergent behaviors cannot be simplified into straightforward theories. Open-endedness, the capacity of a system to continuously generate interesting artifacts, is a key feature of natural evolution. While we have identified necessary conditions for open-endedness, quantifying it remains debated. One case study indicated that human intervention was crucial for achieving open-ended evolution, suggesting that novelty within a specific representation space may be essential. This aligns with the notion that while all interesting things are novel, not all novel things are inherently interesting. Finally, we discuss foundation models, which are large pre-trained neural networks that are transforming various scientific fields. In medicine, they enhance drug discovery by predicting protein folding. In robotics, they automate the design of reward functions, easing a typically tedious process. In physics, large models predict complex systems and are distilled into symbolic equations. AI systems have even achieved high-level performance in solving geometry problems. The potential of using foundation models, especially large language models, LLMs, in A-life contexts has been recognized. LLMs have been utilized as code mutation operators and for setting future goals, but these applications are limited to text-based searches. In our work, we employ CLIP, an image-language embedding model, to align text and image representations. CLIP's versatility allows it to generate images from vector shapes and guide other generative models. We apply CLIP to search for A-life simulations, resulting in analyzable artifacts that offer valuable insights for our research. Section Summary. In this section, we explore various artificial life, A-life, substrates that model complex phenomena, from cellular automata to neural networks, emphasizing their role in studying emergence and self-organization. We also discuss the application of automatic search algorithms and foundation models, like CLIP, to enhance our understanding of A-life simulations through image-language representations. Section: Methods, Automated Search for Artificial Life. In this section, we outline our approach, called Automated Search for Artificial Life, ASAL, which consists of three algorithms based on Vision Language Foundation Models, FMs. Each algorithm employs a unique automated search method to discover artificial life, A-life, simulations. We first introduce key concepts and notations. An A-life substrate, denoted as S, includes any collection of A-life simulations we are interested in, such as all linear simulations. These simulations can differ in their initial states, transition rules, or both. We parameterize S with theta, which defines a single simulation through three components: the initial state distribution, the forward dynamic step function, and the rendering function that converts the state into an image. While we often do not need to parameterize and search for a renderer, it becomes essential when the state values are not easily interpretable. By linking these components, we define a function of theta that samples an initial state, runs the simulation for t steps, and renders the final state as an image. Additionally, we use two functions to embed images and natural language text through the vision language FM, along with a method to measure similarity in that embedding space. A key objective in A-life is to identify simulations where specific events or sequences occur. This discovery helps us find worlds similar to our own or explore possible evolutionary paths, providing insights into the viability of certain life forms. To achieve this, ASAL searches for simulations that generate images matching a target natural language prompt in the FM's representation, allowing us to control which prompt to apply at each time step. Another significant challenge in A-life is discovering open-ended simulations. Finding such simulations is crucial for replicating the continuous novelty seen in the real world. Although open-endedness is subjective and difficult to define, we can capture a general idea of it through novelty in the appropriate representation space. In our work, the vision language FM representations serve as a stand-in for human perception. With this capability, ASAL seeks simulations that produce historically novel images in the FM's representation. Our preliminary experiments indicate that measuring historical novelty yields better results than using variance-based novelty. We also aim to illuminate the diverse phenomena that can arise within a substrate, driven by the desire to understand life as it could be. This illumination is the first step toward mapping and categorizing an entire substrate. To this end, ASAL looks for simulations that create images significantly different from their nearest neighbors in the FM's representation. We find that diversity based on nearest neighbors offers better illumination than variance-based diversity. In the experiment section, we validate the effectiveness of ASAL across various substrates and present new quantitative analyses of some discovered simulations, aided by the FM. Before detailing the experiments, we summarize the FMs and substrates used, with additional information available in the appendix. For our foundation models, we utilize CLIP, which learns general image and text representations by aligning their latent spaces through contrastive pretraining on a large dataset. CLIP provides the necessary functions for embedding images and text. We also use DINOV2, a vision-only model that learns visual representations through a self-supervised framework, but it lacks the text embedding function, making it unsuitable for ASAL's supervised target search. Regarding substrates, we explore several simulations. Boids simulate bird-like objects moving in a 2D space, where each boid is guided by a shared neural network based on nearby boids. Particle life simulates particles of different types interacting in a 2D space, with the substrate defined by interaction matrices and parameters that control particle proximity. Lifelike cellular automata generalizes Conway's game of life to all binary state automata operating on a 2D lattice, with a vast number of possible simulations. Linear extends this concept to continuous space and time, allowing for more complex dynamics. We use the Linear breeder codebase, which defines a substrate with multiple dimensions for dynamics and initial states. Lastly, neural cellular automata parameterizes continuous cellular automata using a neural network to represent local transition functions, with the substrate being the neural network's weight space. Section Summary. In this section, we introduce our automated search for artificial life, ASAL, paradigm, which employs 3 algorithms based on vision-language foundation models to explore various A-life simulations by parameterizing their initial states, dynamics, and rendering functions. We aim to discover simulations that produce desired events, illuminate diverse phenomena, and facilitate open-endedness, using representations from models like CLIP and DINOV2 to guide our searches across different substrates such as Boids and Linea. Section: Searching for target simulations. In this section, we explore the search for target simulations, focusing on both single targets and sequences of targets over time. For single targets, we examine how effective it is to search for simulations defined by a single prompt using Linea, Boids, and particle life. We optimize our approach by applying the prompt after a set number of simulation steps. We utilize CLIP as our feature model and CEP-CMAS as our optimization method. Our findings, illustrated in a figure, indicate that the optimization successfully identifies simulations that align with the specified prompt. Additional results can be found in the appendix. We note that when optimization fails, it often stems from the limitations of the simulation environment rather than the optimization technique itself. When looking at temporal targets, we assess how well we can find simulations that generate a sequence of events using the NCA substrate. We optimize our approach by applying a series of prompts at regular intervals during the simulation. Again, we use CLIP for our feature model and employ backpropagation through time with the Atom optimizer for our optimization process. Our results show that we can successfully find simulations that follow a sequence of prompts. By defining the desired evolutionary paths and using a constraining substrate, we can identify rules that capture the essence of these evolutionary processes. For example, a sequence of prompts like 1 cell, followed by 2 cells, leads to an update rule that allows for self-replication. Next, we investigate the search for open-ended simulations using the lifelike CA's substrate, optimizing for an open-ended score. With a relatively small search space of 262,144 simulations, we apply a brute force search. Our findings, shown in a figure, suggest that the lifelike CAs have potential for open-endedness, with Conway's Game of Life ranking in the top 5% according to our metric. The figure illustrates that the most open-ended CAs exhibit complex dynamic patterns that balance between stability and chaos. We also trace the trajectories of 3 CAs over time in CLIP space, revealing that as we generate novelty, it resonates with human perception. A UMAP plot visualizes all lifelike CAs, showing that the most open-ended ones cluster together, distinct from the main group of simulations. More examples can be found in the appendix. We also use the Linea and Boids substrates to evaluate the effectiveness of our illumination algorithm. A custom genetic algorithm guides our search, selecting parents and creating diverse offspring at each generation. The resulting simulations are displayed in a simulation atlas, which highlights the variety of behaviors based on visual similarity. In Linea, we discover new lifeforms resembling cells and bacteria, while in Boids, we observe flocking and other behaviors like snaking and grouping. Larger atlases are available in the appendix. Finally, we demonstrate that feature models not only help us find interesting phenomena but also allow us to quantify behaviors that were previously only qualitatively assessed. We show different methods for quantifying emergent behaviors in complex systems. For instance, we interpolate parameters between 2 Boids simulations, revealing that the intermediate states lack clear characteristics, highlighting the chaotic nature of the parameter space. We quantitatively support this observation by measuring the similarity of these states to the original simulations. We also explore how the number of particles in particle life affects the representation of lifeforms, finding that a minimum of 1,000 particles is necessary to identify a caterpillar, aligning with the idea that more is different. We quantify the significance of each simulation parameter in particle life by analyzing the standard deviation of the alignment score, identifying the interaction strength between particles as crucial for caterpillar formation. Lastly, we track the change in the CLIP vector over time in a Linea simulation, noting that it stabilizes when the simulation appears qualitatively static, providing a useful criterion for halting the simulation. Section Summary. In this section, we explore the effectiveness of searching for target simulations using various substrates, including single and temporal targets, open-ended simulations, and the illumination of entire substrates, employing optimization algorithms and CLIP for evaluation. We demonstrate that our methods can identify complex behaviors and quantify emergent phenomena, revealing insights into the dynamics of these systems and the importance of simulation parameters. Section: Agnostic to Foundation Model. In this section, we examine the significance of selecting the right representation space by testing different foundation models during the illumination of linea and voids. We utilize CLIP, DINOV2, and a basic pixel representation. Our findings indicate that, as illustrated in the figure, CLIP performs slightly better than DINOV2 in generating diversity that aligns with human perception, while both outperform the pixel representation. This underscores the value of using deep foundation model representations over simpler metrics when assessing human concepts of diversity.